Good morning, ladies and gentlemen. This is Embryology. Sorry about the last presentation. There was no audio, so I'm trying another platform so that you could get an audio for the same slide. We're going to be talking about the second week. This Miss Bobo. So basically, what is happening here is the fourth week of the fourth day all the way to the 16th day, and you can actually see the progression of the size of the embryo from the morula stage day 4 all the way to day 16. So today we'll be talking about basically the second week, which is day 7 to day 14. So but here you could actually see the if you compare the different embryo at different stage. So from the morula stage, which we've discussed, it will soon lose the zona pellucida by hatching, and then it will get segregated. The cells will get segregated into external trophoblast and inner cell mass. So this site is called where you find the cells, they're called the embryonic pole, and the opposite side is called the aembryonic pole. So now you that's called the blastocytes, and then it start implanting. So by day 7 we start by day the end of day 6 we start having implantation, and by day 7 we start having this bilaminar disc. That is what you see that on day 10 you have a blue area and a yellow area. So in day 7 basically you don't have the amniotic sac and the yolk sac, you just have the hypoblast and the epiblast. That is the floor of the amniotic sac and the roof of the yolk sac. But as the days progress, we get to have this amniotic sac, and also we get to have the yolk sac, and below the yolk sac there is like a cavity, that is the chorionic cavity or cilium. And then by the time you head to day 16, which is the third week, you start having 3 laminar discs. You can see the blue cells, then the yellow, then in between them there is a red layer of cells. So down at that point we have the ectoderm, the endoderm, and the mesoderm, as well as the amniotic sac and the yolk sac, and then surrounding them completely is the chorionic cavity. So, scope of today's studies, we're going to see the activities of the second week, which include the formation of a bilaminar, formation of the primary yolk sac, formation of, as well as the amniotic sac, formation of the chorionic cavity, and the extra embryonic mesoderm, which is very important in the formation of the placenta. Formation of allantois and connecting stake, and formation of the primary, secondary, and tertiary villi, part of the fetal part of the placenta. So in this diagram, we get to see the end of the 6, that's implantation. So you see the cells migrating towards the endometrial wall, and it's showing the inner cell, the side that has the inner cell mass implanted. So that is the embryonic pole. The opposite side is aembryonic pole. You can see these are molecules that are being released, which for the BSc student that want to further in embryology, further their studies in embryology, be it masters or PhD, those are just molecules that and cytokinins that involved with some signaling factors as well. But basically what it's saying is just that it needs a place to attach, and that is the point of attachment. And now we go to day 7 where implantation have started. So basically as the cells are implanting, some of the cells, the trophoblast cells, split into 2 cells on the trophoblast and the part that has been implanted. Some of the cells leave their basement membrane and float into the endometrial wall, eroding, releasing some proteolytic cells, eroding the endometrial wall. So you can see them in yellow, that is the cytotrophoblast, and then you can see the syncyte trophoblast, and then the cytotrophoblast holds on to its basement membrane. But at this diagram you start seeing it having projections like a growth, that is to tell you that it has started forming the primary villi, preparing it for the formation of the placenta at this point. Then below here you can see that the implantation has not completely formed. So this is below the 10th day. It is not the 10th day, it might be the 9th, day 8, 7 and 8. A reason because here you've already started seeing the formation of the two layers. You see the yellow cells underneath the light blue cells, so that the two layers are called the bilaminar discs or embryonic discs. And then you could also see the formation of the amniotic sac in above the blue area. So the cells on the upper aspect of the blue cells are referred to as the epiblast, while the yellow one are referred to as the hypoblast. You can see the large empty blastocele, which will soon be lined and form the the yolk sac. Okay, so here you can see that is the tropoblast, that is cytotropoblast, that is syncytotropoblast, and you can see that is the epiblast, the hypoblast. You see the endometrial blood vessel, which will soon be eroded by, you can see how swollen the cells, the endothelial cells of that blood vessel has gone, because the cytotropoblast is planning to erode it. Then you can see that is the primary villi, the formation of multiple layer of cytotropoblast. That is your blastocele, and here H is the uterine cavity. So in this diagram we have diagram A and B. Diagram A is telling us that this is just a few days after implantation, while diagram B is telling us that implantation has completely happened. Has gone beyond day 10, because you don't see the plug in this diagram. And then you see E labeled in the diagram. That E is now the bilaminar disc for the two diagram A and B. All right, so that's A, we have the cytotropoblast in on diagram A. Also in diagram B, A is also referred to as the cytotropoblast. You can see the progression between the two. B is referred to as the cytotropoblast, and you can see it as well here. It has formed a projection, and now the projection has a red structure inside it, which we'll see what the red structure was. Initially you don't see the red structure, we only see several layers of cytotropoblast. That is the primary villi. Here it's called the secondary villi because it has the red substances inside, which is an extra embryonic mesoderm. So see here we are seeing the amniotic sac on both diagram. D is the yolk sac on both diagram, and E is the maternal blood vessel. You can see how swollen it looks like in this B, and F is telling us this is the extra embryonic mesoderm that have formed as the yolk sac is reducing in size. And you can see it in diagram B, it has split into two. G is the splanchnic extra embryonic mesoderm, H is the somatic extra embryonic mesoderm, which is associated with this cytotropoblast, and it will grow into the cytotropoblast to make this the projection, that primary villi into a secondary villi. In between them is the chronic cavity or coelom, and you can see the mesoderm, the somatic mesoderm, going into the primary villi. So now it is no more referred to as the primary villi, but rather it is referred to as the secondary villi because it contains the mesoderm. And then you can see that the mesoderm splitted completely all around the forming embryo except at K, and that K is called the connecting stage, which will form the future umbilicus. And here this is few days afterwards, after the diagram we seen initially in the previous diagram. So here you can actually see the other structures as well. You could see the allantoids as well. You could see the lacuna around the forming embryo. And so A A is amniotic sac, B is yolk sac. In between the two, you can see a layer there that is telling you that we have gone beyond the second week. We are in the third week in this diagram, but just for information's sake, we are now having a detailed class about the third week. You can see a mesoderm in between. C is our in cytotrophoblast, D is cytotrophoblast. At this level, it's still showing you it's a primary villi. It has not been filled with the extra embryonic mesoderm. Here you have the connecting stick, and then here F is a lacuna. The space is found in between the cytotrophoblast. It will soon be filled with space and maternal blood that comes from the eroded blood vessels. And here is an eroding blood vessel. You can see the blood cells moving into the closest lacuna. And H, you can see that is the coronary cavity, and you can see I is the mesoderm. You can see J is that our smooth or somatic extra embryonic cilium, which grows into this villi projections. In this diagram, it is just comparing what we've discussed in four stages of embryonic stage. You can see implantation through the embryonic pole, and then you can see how the cytotrophoblast is eroding the endometrial wall. You can see how it is forming lacuna around, and then you can also see how we start having the formation of extra embryonic mesoderm and also the coelom. This diagram also is the same thing, comparing what we've seen earlier on. So here you can also see in the first diagram, day 7.5, you can see we have two layers, and also you have the amniotic sac and the yolk sac. Day 12, you can see that we have primary yolk sac. Look at the size of the yolk sac, it has reduced, so now it is called a primary yolk sac. And then you can see the surrounding red area, which is the extra embryonic mesoderm, which will soon split into two. In the third diagram, day 16, you can see some of the mesoderm are surrounding the embryo, which is called the smooth extra embryonic or splanchnic, and some of them you can see them going into the villi formation and around this line the cytotrophoblast and going into the cytotrophoblast, and that is called the somatic extra embryonic cilium. The space between them is referred to as the coronary cavity, and you can see the connecting stick, and then you can see another projection of the yolk sac going into the connecting stick, that is called the allantoids, which is also important and the formation of the urinary bladder in the future. Here on same diagram, the third diagram, you could also see blood from the eroded maternal blood vessels going into the lacuna, and this lacuna are important because if you notice this the cytotrophoblast will soon grow above the cytotrophoblast will soon grow above cytotrophoblast in this diagram. In this the diagram above on the right top corner is just to show you how the epiblast and the hypoblast are sitting down there, and then you have a pre-caudal plates, which is very important we're going to talk about it in the third week. And here we have stages of placental formation. Just like the other diagram, you could also see cytotrophoblast eroding the endometrial wall. You can see the blood vessels. You can see the size of the amniotic sac versus the yolk sac, and gradually you can see the mesoderm surrounding it. You can see that blood is now found in the lacuna in B. And when you go to C, you can see the embryo has the size of the embryo has also reduced. You have a 3-laminar disc in C, and then you can see the projection going into the cytotrophoblast. You can see the blood entering the lacuna. By the time it is D, you can see that the blood has filled in the lacuna, and the mesoderm found inside the cytotrophoblast have become blood vessels. Alright, so at this stage the villi are no more primary or secondary, but rather they are now called the tertiary, because the mesoderm filled mesoderm have differentiated into the blood vessels. At E is just showing us the formation and position of decidual cells. What happened is that immune cells gather around the junction between the cytotrophoblast and the endometrial tissues, and changes the collection so that the endometrial tissue around the embryo specialize and form a protective immune layer that permits this foreign object, because the embryo has an DNA from the father, so it has a foreign DNA in it. So this slide is also, A is telling us the same story we've been seeing. You can see from implantation, you can see blood vessels from endometrial wall, and you can see the growth of the cytotrophoblast forming the primary villi. In B, you can see the size of the yolk sac. You can see the extra embryonic mesoderm around it. You can see the lacuna also, and the blood vessels trying to empty into the lacuna. In C down here, you could also see how the the cytotrophoblast has formed projections, and you can see mesoderm has filled the projections. So at this point, it is not a secondary villi because it has mesoderm within it. And then if you go to diagram D is showing us that at this point it's just the cytotrophoblast has grown. You can see inside the darker cells are the cytotrophoblast, the light blue outside is the syn cytotrophoblast. Alright, so in this it is called the primary, and in E it has gone beyond the secondary because you can start seeing blood vessels. So the moment this pink area within the growing projections, that is when you have a secondary villi. But in this, the blood vessels have started forming, so at this point it has started becoming the tertiary villi. And by F, in both that side, you can see that in F you have some of the cells of the cytotrophoblast going to anchor themselves above the syn cytotrophoblast. So they've grown beyond the point of the syn cytotrophoblast. So they encircle the lacuna and the syn cytotrophoblast. Inside them, they contain capillaries, blood capillaries, which come down and connect to form the umbilical artery and vein. So at this point it is referred to as a tertiary. As well, if you look at the diagram F on the left hand side, you can actually see that these purple cells here are cytotrophoblast. Okay, they have grown beyond the syn cytotrophoblast because the syn cytotrophoblast is the blue one around it, and you can see that syn cytotrophoblast cells are growing beyond it. And here you have the blood vessels of the maternal spiral artery emptying into the lacuna. So you can see that now the lacuna containing the maternal blood, all right, and the villi, all are found in between the syn cytotrophoblast from the lower aspect to the one anchoring it. So you can see that the villi have branched into different branches. So the one that goes up all the way and goes beyond syn cytotrophoblast is referred to as anchoring, while the ones branching are referred to as the branching villi. So in this diagram, we still explain the first one is the primary villi. You can see outside the orange color referred to as syn cytotrophoblast, and the inner one is cytotrophoblast. So that is primary villi, and then you can see the longitudinal view. And then the second one is secondary villi. You can see outside you have the syn cytotrophoblast. Then you have the cytotrophoblast. Inside you have a mesoderm. All right, so you can also see the longitudinal view of it. And so here you still have a cross-sectional view of the tertiary. You can see outside you have syn cytotrophoblast, you have cytotrophoblast. Inside where you have the mesoderm, you can see blood capillaries forming, or blood vessels. So basically that's what is indicated here in this diagram. So you can see that the first one is a blue one. Outside that is syn cytotrophoblast. It has formed. The black area around it is called the lacuna filled with a maternal blood. Okay, that is where you have the exchange diffusion. All right, so you can look at the green area also, that is your cytotropoblast. Look at it going all the way to anchor itself onto the decidual cells. Is going to grow beyond. So in the third layer, on the second branch, you can see the green structure going beyond the blue. So at this point, it has formed the anchoring, while the other ones that have not reached the deciduals is called the branching. All right. So down here you can see the chorionic plates, which is the mesoderm, which is the extraembryonic mesoderm. So in this diagram, you could actually see the progression of the villi, and you can see that at this point you have villi all around the embryo, but at a particular week you don't have villi at the opposite end of where you had the first implantation. All right, so you get to lose the villi on the opposite side. So here is still formation of the villi. So here what you get to see is external. On the first diagram you can see a yellow structure completely. So the outer yellow layer is cytotropoblasts. You can see it all the way to the pink area where it has the endometrial wall. So the endometrial wall, you can see there are two layers. You have the myometrium, which is the upper part with the maternal blood vessels. Then closer to the yellow structure is like a light pink, and that is the decidual cells. So you can see what happened in the decidual cells. The cell have become immune protectors of these particular villi or the embryo completely. This is such that they don't kill this embryo because it has a foreign DNA in it. So here you have externally, you have the cytotropoblasts. Around cytotropoblasts, you have the lacuna, which is filled with maternal blood. Then you have the cytotropoblasts, and then inner you have the mesenchyme mesoderm or the chorionic plate or the extraembryonic mesoderm, and then you can see the blood vessels growing. Inside it are capillaries, but they fuse together to form one blood vessels inside the chorionic plate. That is referred to as the umbilical artery or vein. And here also is just showing us the same thing. So here you could actually see the purple area is referred to, the outer purple layer is referred to as cytotropoblast. You can actually see surrounding empty space, which is called the lacuna, but it's not actually empty in real life. It has the maternal blood there, so you only have diffusion between the capillaries of the embryo within the villi and the maternal blood found in the lacuna. So you can see how it has grown all the way to anchor itself onto the decidual plate, and then you can see that you have the cytotropoblast anchoring the villi on the decidual cell. So the cytotropoblast has grown beyond that, and inside you can see the capillaries forming. At the bottom here you can see major artery and vein. These are what is referred to that form the umbilical artery and vein. So as that is forming, we have the formation of the extraembryonic membranes as well. So here on the first diagram, that is cheek. We don't need to explain cheek, but here in the human you can see the pink layer surrounding it, and then you have the villi at one point. What surrounds it completely outside the pink is called the chorion. All right, that's the chorion. It's made of the placental layers. Okay, the chorion is made of placental layers. You have the capsularis and parietalis. That forms the chorion from the outside. Then inside, next you have the amniotic sac. You can see the embryo also lying inside the amniotic sac. So between the chorion and the amniotic sac, you have the chorion cavity, and then inside you also have the yolk sac, which has become shrinked. And also you remember our allantoids. The allantoids is also there. All right, so and this point where the bottom point, you have also the villi. You can see the villi here, and then the pink area or the reddish area is indicating the maternal part of the placenta, which is formed by the decidual cells. This third diagram is also depicting what is found in the second diagram, so I don't think I need to explain that. So here we are going to understand how the different parts of the decidual cells. So if we check from diagram C, D, E, and F, that this layer is referred to as the so the white area outside the embryo here, sorry, the gray area, not the white. This arrow is pointing to the white, sorry, it is the gray area. That is decidual capsularis because it's encapsulating the embryo. The next one is decidual parietalis because it is around the muscle area, the remaining part of the uterus, so it is called decidual parietalis. And then you have decidual basalis, which is found at the base of the embryo. You can see that part where I have the villi placenta, so that is it. As the embryo continues to grow, what happens is that the decidual capsularis and parietalis will come and fuse together and they form the chorion. So in this diagram, you're seeing that the embryo, you can see the extra embryonic membranes as well. You can see the yolk sac, you can see the amniotic sac, you can see the growing embryo. This is around the 6th, 7th week because you start seeing the upper and lower limb buds growing. You can see how it is completely surrounded by blood vessels. Sorry, it is around the 4th week. So because the 6th week is ossification, sorry, so this is 4th week because the upper and lower limb buds are forming. Ossification is 6 weeks, and so you can see that the placenta is completely surrounded by blood vessels. You can see the maternal or the decidual blood vessel, the spiral artery emptying into the lacuna. So in between this embryonic blood vessels, the blue and red vessels, you see space that are filled with red. Those are the spiral artery emptying into the lacuna, which is a point of diffusion between the embryo and the mother. So here you can actually see that. So you can see that you have the decidual capsularis and you have decidual basalis, parietalis just beyond it if you check very well, and above that area is also decidual basalis, and you can see the embryo inside the amniotic sac and then inside the chorionic sac again. So applications of the placenta, it's really important because this diagram you can see that if you can see the size of the embryo, you can actually take amniotic fluid to test for genetic materials in Western countries because they are having so much congenital defect and they don't want to have so much expenses. What they do is they actually could take the sample of the amniotic fluid and test if that embryo is okay, if they need to give birth or they need to terminate the pregnancy. And also you can take biopsy, you can take samples as well from laparoscopy and test it if the mother has like a viral transfusion that might be going into the embryo to cause a particular congenital defect like the cytomegalovirus. Another one is the size of the spiral artery, the terminal part of the spiral artery emptying into the lacuna. If it narrows, then you might have a tendency to have a prenatal eclampsia, which is terrible. So it has to be open whilst to have a normal pregnancy. And this diagram is just summarizing what we've talked so far from day 1 all the way to having a bilaminar disc and also the formation of the extra embryonic membranes. Here is just to compare the different stages of the formation of the placenta. I hope you do appreciate it, and thank you very much. If you have any question, you can share it onto the link. Thank you. Whew.